everyone, this is Perry. Today I want to show you an article that's named The Seasonal Variation in Biomarker Responses of Donax Trunculus from the Gulf of Onnobah, Implication of Metal Accumulation in Sediments. Firstly, I want to give you a brief introduction of the article. The aim of this study is to test the biomarker's responses in an edible mollusk, Donax trunculus L, which is associated with environmental pollution in the Gulf of Onnobah. And the biomarkers selected were glutathione S-transferase and acetyleninesterase and methylencyl. And the sample collected seasonally in September 2014 and January, April, and July 2015 from two sites located over the Gulf of Onnobah, which we see is the L and the Sadie. And why this study chose the Donax trunculus? Firstly, I want to give you some brief information about this species. This is a abrupt wedge shell or wedge clam and is a bivalve species. And the bivalve belongs to the first choice species as bioindicators for environmental and chemical stress. And they are sentinel benthic organisms living as filter feeders and exposed to different environmental compartments. And due to their wide distribution, resistance to variable environmental conditions, which predispose them to the direct absorption and accumulation of a wide spectrum of waterborne chemicals, so they are prolific tools for the biomonitoring of chemical pollution. As well, they have the possibility to measure several biochemical, cellular, and physiological biomarkers, makes bivalve suitable organism for investigating the effect of chemical pollutants. And why this study use these three as biomarkers? Firstly, for the GSTS, which is a multiple enzyme family involved in phase 2 detoxification processes and are used as biomarkers of exposure to several groups of pollutants, including some pesticides and PCBs and some petrochemical products in invertebrates. And ACHE is involved in the hydrolysis of neurotransmitters acetylcholine. And the measurement of ACHE inhibition in marine organisms has been widely used as an indicator of environmental contamination by organophosphate and carbamates pesticides. And for the MTIs, which are low molecule weight and cysteine-rich cytosolic proteins able to bound to many metals such as Ag, Cd, Cu, and some others. And the MTIs are involved in different biological processes like the homeostasis of essential metals, detoxification of toxic metals, and cell protection against oxidative stress. And why we measure metal content in sediments? Firstly, because the sediments are an important sink for metals and other pollutants. The benthic invertebrates are known to take up and accumulate metals, both essential and non-essential, and from water and sediments as well as from their food supply. In addition, the concentration of heavy metals were determined in sediments originate from two sites, the L and the Sadie. And now I want to show you the materials and the method. Firstly, is a study area, the Gulf of Onnobah, and one of the site is the L site, which is located about 30 kilometers to the east of Onnobah, far from any human activities, and is considered as a relatively clean sites, so we think it's a reference site. And the city site, which is located about 1 kilometer to the east of Nova City, it is receives industrial and domestic wastewater and is considered as a polluted area. For the sample collection, the molecular bivalves, the D. truncularis, the sample was collected same range shell length, and in the autumn of 2014 and in the winter, the spring, and the summer of 2015 from two sampling sites, the L and the CD, and transferred to the laboratory. And the sediment samples also collected at these two sampling sites in the winter 2015 for meta-analysis. And for the biomarker analysis, the mantle of 6 bivalves was dissected and samples were prepared for biomarker analysis. Firstly, for the GST activities was measured using 1-chiral-2,4 dinitrobenzene as substrate. And as for the specific measure of this environment, we can see in the Habig studies, and the activities rate was expressed as mmol per minute per milligram protein. And for the ACHE activities was measured with the use of acetylcholine as substrate, and the activities was expressed as mmol per minute per milligram protein. And the metal and for the MTS level, the mental samples were homogenized in 3 volumes of 20 mm trisulfurous buffer with 0.1% of beta mercaptan ethanol and 0.5 mm of PMSF, followed by ethanol chloroform extraction after incubation with DTNB, and the level of MT were calculated assuming this relationship shown in the slide. And for the metal analysis in sediments, the total metal content in dry sediments were determined by atomic absorption, spectrometry after high pressure microwave digestion with nitric acid. And for the statistical analysis, the comparison of the mean value between sites was made by t-test, and the combined effect of seasons and sites were investigated using ANOVA, and the difference were considered as significant when p-value less than 0.05. And here is the result and discussion. Firstly, for the GST, in this study, the higher activities of GST observed at L in autumn, which is probably associated with the restoration of reserves and the period of sexual rest and the food availability in this season. And in SETI, the GST activities was lower in summer. This decrease might be related to the spawn period of D. truncularis, which means the sex and the reproductivity period is related to the GST level, and also the significantly higher GST activities were recorded in bivalves from SETI compared with the L. And the site of SETI is exposed to the waste products from complex pollution, which we think this significant difference is due to this reason. And for the ACHE, the seasonal variation of ACHE activities in the mantle of D. truncularis showed a decrease in autumn and winter compared to spring and summer for both these two sites. And the variability of the ACHE is not directly related to the age, the sex, the reproductive period of the organism, but to the seasonal variation of temperature, especially to the seawater temperature. And the inhibition of ACHE was detected in bivalves from SETI compared to the L, which shows the metal inhibit ACHE activities. And for the third graph, the empty concentrations in D chunklers collected from the ANOVA Gulf varied between each season, with higher concentration in summer and lower in spring. And the basic level of MT in organisms have shown to alter with the season, the reproductive state, and the water temperature. And the data showed an increase of MT level in this species from SETI compared to L, which recorded the highest value in summer. And here is a result and the discussion about the metal concentration in sediment. We can see from this table, the Gulf of ANOVA remains contaminated by metals, and for the ME, this metal is higher in the L sites, which is because this is an essential metal, and also showed an significantly higher concentration of CD, Cu, Pb, and Zn at SETI than the L, and there is no significantly difference between the two sides for Fe. And here is the conclusion. Firstly, the biomarkers measured in the mantle of D trunculus, which is a useful sentinel organism for environmental monitoring, and the analysis of mantle using these three biomarkers in sediments confirmed the metallic contamination of Gulf of ANOVA. The level of metallic pollution was found relatively high at SETI compared to the L, which is result from the biomarkers graph and the biomarker analysis. And the result will contribute to the development of an environmental monitoring program based on relevant biomarkers. And here is some questions. Firstly, why is it necessary to observe parameters of the biomarkers both in different seasons and in different sites at the same time? The second is, besides these three, are there any other metal biomarkers? The third is, why do you think metal biomarkers are important for environmental monitoring? And the fourth is, what limitations does this article have? So I'm very happy to hear from you. So thanks for your listening.